Hi, John, you can hear us?
Yes, I can. Does that look okay?
Yes. So then, yeah, it's nicely, presented here. So then, are you ready now?
Anytime, Makoto.
Sorry?
Yes.
Yes. Anytime you said. Yes, so then now, yes, I introduced that just briefly. 皆さん来てくれてありがとう。今日はね、sorry that I have some interaction with the floor, some in Japanese. 授業にもしているので、授業とか、あるいは、まず第一にですね、大学とはあんまり関係のない、市民講座として来られている人どれくらいいますかね。大学と関係ない人、うちの大学と関係ない人手を挙げて。Yes. じゃあ、私の授業の人はどれくらいいますか？手を挙げてください。はい。それで、あとは、大学の多学科の研究科の人はどれくらいいますか？農学研究科の人ね。はい。それで、じゃあ、生物学科の4年生以上の先生も含めてどれくらいいます？わかりました。生物学じゃない人はいますか？バックグラウンドが生物学ではない人。Yes. So then the good distribution of the audience. The half are the undergraduate people, and half are somehow the graduate school and the faculties, and who are working on biology. And some people are not so much familiar for sometimes they should be familiar for the cell biology, but not so much about protein, the molecule, and the genes, genetics. Yes. And yes. And I just very shortly I introduced the Professor John Glass. The John Glass is he is the long time of the really good friend of us. He used to work on the mycoplasmology. But he worked with John Glass Benter. So then he said more over the 20 years, and he's been taking care of the John Glass JCVI. I mean the Craig Benter Institute, the big project. So then how to decide determine the first genome and synthesize the whole genome and transplantation of the genome. When finally they achieved making the minimal bacterium, JCBI Sin3. So it was everything anytime. So then his and their work was really impressive for us mycoplasmologists, and as well as all the biology people. So then I think that he will introduce briefly by himself. So then I don't say too much. So then, and also the big thing is, the Professor John Glass was scheduled the plan to visit on site us, because we did kind of the big international meeting in the Chuo Kokkaido. Then it's really interesting place that you think about to do some activity by yourself. You should think anyway. And unfortunately, he got suddenly he need to get kind of the big surgery. So then now that he is still on the bed, but he said we worry about that it's maybe the very stress, the big stress if we ask him Zoom the lecture. But he said that he's getting very quickly. They're getting they're recovering very quickly. So then it's okay to give the real time lecture on Zoom. But then John, what time is it now?
It is 7:24 p.m.
PM. So that's a good time. Good time for activity. Yes. So then now so then please start. So then we look forward. So then John Glass lecture. Please go ahead.
Makoto-san, thank you very much for inviting me to do this And this is such a pleasure. And I am so sorry that I can't be there. Is my volume loud enough? Am I speaking loudly enough? Can you tell me yes or no? Is this too loud, not loud enough? How is the volume? Is my sound okay? Makoto is.
Okay. Now we are doing now. Sorry that we forgot it.
Okay, here we go. So I am John, and I have been a mycoplasma person for 30 years now. And it has been one of the great joys of my life. And I have known Makoto, I think Makoto-san for at least 25 years of those. But I want to tell you a story about that I have been working on for the last 25 years. And I'm speaking for myself, for the JCVI, as well as for members of the Minimal Cell Research Consortium. And I have loved this cell and been working on it so long that I think of it as being like my family. And so I was just going to show you, before we really get started, some of what I think are beautiful pictures of my children. So hang on, let's see, here we go. So here is a scanning electron micrograph of the minimal cell. So this is a clump of hundreds of cells showing their various sizes. And here, this is just an astonishing photo made by your own Makoto Miyata and his graduate student, Hana Kikiyama. And it is one of my minimal cells that he has installed 5 spiroplasma genes to make this normally non-moving organism swim around, which is just amazing. And then we've got some high-resolution microscopy showing dividing cells where you can see the DNA and the cytoplasm and the membrane. Here we've got a negative stain EM of the minimal cell. Of course, mycoplasmas make these colonies that look like fried eggs, which I think are so cool. This is a cryo-electron tomogram of the minimal cell, and you can see on the right that we've highlighted the ribosomes. So we were able to count 700 ribosomes in a single cell. And then I have computational biology colleagues who have made a computational model of our minimal cell, here showing proteins, DNA, ribosomes, the little yellow balls. But this is just a remarkable thing that we will talk about as the talk goes on. And then for me, this is one of the most amazing moments in my scientific career, this showing how bizarre, strange cell division by the minimal cell that we'll talk about in a few minutes. But this is the minimal cell. I would get a tattoo if I was such a person, and maybe actual size though. Okay, so our team started working on this project really before I ever joined the Venter Institute back in the early 1990s. But in 2010, we reported in the journal Science that we had made a near wild type version of the goat pathogen Mycoplasma mycoides that had a synthetic genome. And it was called the world's first synthetic cell. And this was JCVI Syn 1.0. And then 6 years later, we built the world's first minimal bacterial cell, which we call JCVI Syn 3.0. And this is, I'm going to talk to you about the story of building this cell and what we have done with it. So first, we published this in 2016, and it was amazing. Now, more than 60 academic and industrial labs have come forward wanting to be part of our community of folks who are studying this minimal cell and using it both for applied purposes and to try to investigate the first principles of cellular life. Why did we do this? the question that has driven me and many of my colleagues is this question of what is life? And despite all that biology has learned in the history of biology, we really don't know that much about exactly what it is to be alive. What's the difference between a dead cell that is a bag of chemicals and a living cell that divides and metabolizes? Now, my team, we are genomics people. We work with DNA. And so we wanted to use the lens of genomics to ask questions such as what genes are necessary for life? Or maybe putting it another way, what tasks must a living cell perform? And I would like you to think of, so in the 19th and 20th century, physicists and chemists used the hydrogen atom to understand the basic principles of matter. There were things they could do with the hydrogen atom that we still can't do with more complicated atoms, but they always and have since proved that what is true for the hydrogen atom is also true for more complicated atoms like oxygen, lead, uranium, carbon. And so I would like you to think the way we do, which is I would like the minimal cell, because of its simplicity, because we can do things with it that cannot be done with more complicated cells. Think of this as the hydrogen atom of biology. And I'll try to show you over the next hour why this is so. So what is a minimal cell and why are we interested in this? A minimal cell contains all the essential genes needed for independent growth under ideal laboratory conditions. No gene can be removed without a loss of viability, and it has all the machinery needed for cellular life. So the idea is that if we understood the function of every gene, it then might be possible to obtain a complete understanding of what it takes to be alive. And it might also then be possible to do computational modeling of what first the minimal cell is and then more complicated cells. And with this, we can predict the effects of environmental variation or by adding metabolic pathways and really start to think about understanding maybe eventually what every atom in the cell is doing. Now, I worked with, I know there are people who work on trees that, have 50 years in order to see a tree lifespan. And I used to work on a bacterium that made a colony every 6 weeks, Mycoplasma genitalium. So we decided that we wanted our cell to be the minimal cell. It had to have all these characteristics, but it also had to divide every 2 hours. And we couldn't take any genes out without substantially slowing the growth rate. So the target we chose is an organism called Mycoplasma mycoides. For those of you who are not familiar with mycoplasmas, mycoplasmas are atypical bacteria. They evolved from more complicated bacteria like Bacillus subtilis or E. coli or Staph aureus, but they live in such stable environments like a respiratory system or a urogenital system that they were able to throw away a lot of genes. So Mycoplasma mycoides is a gram-positive, very small bacterium. You can see here the mycoides genome or cell drawn in relative to the size of an E. coli or a bacteriophage. It is a pathogen of goats. Probably it needs all of its genes to infect a goat, but we wanted to know what genes were necessary for growth in laboratory media. It's already nearly minimal in the sense that it has a very small 1 megabase genome and makes only a little under 1,000 genes. It has minimal metabolic complexity. It is a total heterotroph. It makes nothing. No amino acid, nucleotide, or fatty acid biosynthesis. No cell wall, and you have to grow it in a very rich growth medium. And it makes these beautiful colonies that look like fried eggs. In this case, the cell has a LAC-Z gene, and so it's grown on agar with XGAL. It has about a 60-minute doubling time. How did we build a bacterium that had a synthetic genome? We started with oligonucleotides. And so to build a genome, we take overlapping oligonucleotides and assemble those first into cassettes that are 2,000 to 7,000 base pairs. These are then assembled by homologous recombination. The edges of these cassettes overlap, and so they can be assembled in vitro. And then we would build the thing in vitro and then install it in a yeast as a yeast centromeric plasmid or a yeast artificial chromosome. So our genome here has about 1 million base pairs of mycoplasma sequence and about 3,000 base pairs of yeast vector sequence. The yeast sequence means nothing to the mycoplasma, and the mycoplasma sequence means nothing to the yeast. The next technology we had to develop to build our cell was called genome transplantation. And here we have to take this clone, this yeast artificial chromosome, and install it in a different species of mycoplasma so that it takes over the cell to produce a new cell with the genotype and phenotype of the synthetic cell. Now, our goal was to try to learn what cellular software was necessary to perform the tasks of life. And so we made a list based on what we understood about life as to all the things that living cells have to be able to do. And for instance, living cells have to be able to have lysine, the amino acid. And some cells may have 20 or 30 genes that allow them to synthesize lysine. Our mycoplasmas cannot synthesize lysine, so they have one transporter protein that can supply them lysine from the extracellular environment. Both ways are totally effective ways to perform the task of getting lysine, and it's just different numbers of genes required to get there. And so we wanted to figure out how few genes and what the genes were that were necessary to make something that's alive. And so again, it doesn't matter what genes enable the completion of a task needed for life as long as they are present. The way we went about genome minimization is something you hear about in engineering and especially in synthetic biology. This idea of design, build, test. Sometimes it's design, build, test, learn, but it's a cycle that here we use it for genome minimization. And so, I'm sorry, back. First, we wanted to synthesize a genome, build the thing, test it using genome transplantation, and then learn and redesign it and go again as needed. So the design, there were heroes of this. Two of my colleagues, especially Clyde Hutchison and Hamilton Smith. So Hamilton Smith won a Nobel Prize for the discovery of restriction enzymes, and Clyde Hutchison was one of my professors in graduate school, and twice has been on the list of people to get Nobel Prizes, but didn't quite, they only have 3 people who get the prize, so he didn't. But these guys are amazing scientists, along with JCVI professor Chuck Merriman. They had the main task of designing the cell. And so to do this, we had 2 approaches. So one way would be to keep taking out genes, sequential deletions. You delete a gene and you see, does it make a living cell or not? And you characterize the cell. This is how several groups have minimized E. coli, for instance. But what we wanted to do was a different approach. We would design it based on a knowledge of what genes are essential and what genes are not, then synthesize the genome from chemicals, assemble it, and build a bacteria with the reduced genome. The way we learned what genes were essential as opposed to what genes were non-essential, is we used transposon bombardment. Transposons are pieces of DNA and in combination with an enzyme that allows that piece of DNA to jump into, to integrate into a genome, a bacterial genome. And so we had this little piece of DNA, less than 1,000 base pairs, that had a tetracycline resistance gene in it. If this jumps into a gene that is essential for life, that cell will die. If the transposon jumps into a non-essential gene, that will produce a living cell that is resistant to the antibiotic tetracycline. And then we can sequence out from that antibiotic, from the transposon, to identify the site of the transposon integration, or what gene it integrated in. And so the way we do this, we used a transposon called TN5. We have a piece of DNA that I think is about 100 and, I don't know, it's a few hundred base pairs long. We mix that with a commercially available enzyme, transposase, to make the transpososome complex. We then introduce that to a culture of Mycoplasma mycoides by polyethylene glycol transformation. And this will, when you plate this out on agar media in Mycoplasma growth media, we got about 80,000 colonies. And so this is passage 0. We then take those colonies and put them into fresh growth media. That will be passage 1. And then we do multiple cycles of put cells into growth media, grow it for a day, then take a small aliquot of that, move it on to passage 2, passage 3, and passage 4. So only the fast growers will be represented in passage 4. We then, so passage 1, 0, 3, and 4. Next, we isolate the DNA from these passages and cut them with restriction enzymes. Then we sequence out using primers that bind in the transposon. And this lets us identify the sites of the transposon integration. So if you look at a piece of the genome here, where the genes are in the dark black marks. So first you see, and we have the white boxes over the genome are library from passage 1, and the red boxes are over passage 4. So essential genes have no transposon insertions. You don't see any of the boxes over this gene, CTP synthase. Whereas a gene that causes the cell to grow more slowly will only be hit by the transposons in passage 1. And you see there are only white boxes over this protein of unknown function. Whereas a truly non-essential gene will have both red boxes and white boxes, because it doesn't reduce the speed of bacterial growth. And so what we found is about half of the genome is either quasi-essential or essential, and about half is non-essential. So based on this, we decided how we would synthesize a genome. And we produced what we called our first hypothetical minimal genome. Hypothetical minimal genome. And it was about half the size of the original Mycoplasma, the original JCVI SYN 1.0. And we made the genome in about eight subsections of equal size, and each of these overlapped each other. So the first thing we did is we tested genomes that were 1/8 minimized and 7/8 wild type. Now, our first effort was done too fast, and only one of the 8, 7/8 wild type, 1/8 minimized, grew, but that still told us that this was going to work. Then we went back in and did much more transposon bombardment in much greater depth. So we were much more certain about what was essential and what was not. And this led us to produce the reduced genome design, RGD 1.0. So if the wild type organism is 1 million base pairs and you see 8 overlapping pieces, the reduced genome design was a little over 500,000 base pairs. And this was a 50% reduction. And the building was done by 3 of my colleagues who were primarily yeast geneticists. Those of you who do molecular biology may know Dan Gibson if you've done Gibson assembly. And so the way we did this was outlined in the paper we published in 2010. Basically, the first step was in vitro. We synthesized, and we had this done commercially. We developed the methods for synthesizing from oligos, but it's just not efficient labor for us to do this. So we purchased small genome fragments, 1.8 kb, and then did error correction of these, PCR amplified those, and then transformed them into E. coli to produce small fragments that have 5 of these 1.4 kb fragments that were about 7,000 base pairs. And we would sequence verify those in E. coli and then move those into yeast by transforming yeast. And eventually we're able to build molecules that were 1/8 of the intended genome, and then finally the complete genome. And so again, we did it in an 8P strategy. First, we synthesized genomes that were 1/8 minimal and 7/8 wild type. And the next thing is we gave those to our genome transplantation team. Now, genome transplantation is the key. I was always sure we would be able to synthesize DNA. We did it much faster and better than I expected, but this we were not sure if it would ever work. Carol Lartigue was the scientist who spent 2 years working night and day to do this, and finally it worked. So this is how we think it works. Imagine that you have the red genome that has a tetracycline resistance marker in it. And we transform that by adding polyethylene glycol and calcium to a group to Mycoplasma capricolum cells. And capricolum is very genetically similar. It's another goat pathogen, similar to Mycoplasma mycoides. And just like you're doing a chemical transformation of E. coli, the genome somehow makes it into the cell. Then we return the cell to growth conditions by moving it out of the transplantation reaction into bacterial growth media. And cell growth ensues, and you get cells that are transiently diploid. They divide, and then you add tetracycline to the growth medium, and that will kill the cells with the wild type genome, leaving you only with the cells with the synthetic genome because they are resistant to tetracycline. And to our enormous delight, we got for all 8 of the cells that were 7/8 wild type and 1/8 minimized, they were all viable. So here you can see scanning electron micrographs of those. What we also found is that the complete RGD, reduced genome design 1.0 minimal genome was not functional. However, if we mixed the wild type 8 pieces and the minimized 8 pieces, we got genomes that were partially mutated. And we found that some genomes that were as much as 5/8 minimized still grew well. The colonies may have been a little small, like you see in the red box, but they're still there. Now, we realized that they're there, they're growing slowly. And we also figured out that the problem we decided was synthetic lethals. And for instance, so what's a synthetic lethal? Synthetic lethals are pairs of genes that perform an essential function. Neither gene is essential. So for instance, we have two copies of the ribosomal RNA operon, the RNA, the genes that make the RNA that go into ribosomes. And we knew that the cell only had to have one of them. And transposons were constantly hitting one or the other one. Those are easy to figure out. We knew we had to either leave both of them or one of them. But if you have two genes that both do the same thing, and both of them are essential and both are non-essential, you might take both of those out and you would get a cell that was not viable because you have to have one of them. So this led us to go to reduced genome design build test cycle 2. And for reduced genome design 2.0, we used a cell that was half minimized, RGD 1.0, that had 4 minimized strains, and half wild type, 4 wild type strains. And then we did transposon bombardment again. And we found that 26 genes that were previously non-essential in the wild type segments, 1, 3, 4, and 5 became essential or quasi-essential. So then we rebuilt RGD 2.0, and that produced a viable cell that was about the same. It made about the same size colony, and it doubled almost as fast as the wild type organism. Kim Wise, a famed mycoplasmologist for the last 30 years, did this work. And the colony morphologies are the same. The growth rate is about the same. But Craig Venter asked us if we can do one more cycle to try to get rid of some more non-essential genes, which were still in the genome. So we did RGD3, and we moved 37 known non-essential genes. We reduced the size of the yeast vector. We moved 1 RNA operon, and we enabled to get a reduction to produce a genome that was smaller than any cell that we know of that can grow in pure media in nature. RGD was built, it's viable, and this we call JCBI Syn 3.0, a complete 531,000 base pair genome. We were just thrilled. This was one of the great days of our lives. And here's a scanning electron micrograph of the cell. The funny surface, I think, is an artifact of the EM fixation process. And so for the rest of the talk, I'm going to talk to you about analysis of this thing. Again, here's what the cell looks like, but I want you to think of it differently for a bit. Think of it as a salad. And this is how we're thinking about this salad. So what's in this salad? Well, about half the genes are involved in the central dogma of molecular biology, expressing genes, making proteins, preserving DNA. Now, surprisingly, we were not expecting this to be so large. A large number of genes we had no idea what they did. We didn't expect this, and we'll get to that in a moment. In fact, we'll get to it right now. So this is complicated. So here are, I think, 13 lines. Each line corresponds to the genome of an organism. So it goes mycoplasmas of the same type as our minimal cell, other types of mycoplasmas, bacteria in general. So Bacillus subtilis, Staphylococcus aureus, E. coli, Thermatoga, no, not Thermatoga. Is there not? Yes, Thermatoga maritima, tuberculosis, Homo sapiens, humans, fruit flies, yeast, Arabidopsis. And we took the proteins that were encoded by Syn 3.0, and we used a technique where we compare the sequence of the proteins with the sequence of the proteins in each of these organisms. So imagine for E. coli, we take protein 1 of the minimal cell, and we do a blast search of the E. coli genome. And if there is a protein in E. coli that is similar to the protein in the minimal cell, we put a blue box in this, on this line. If there is no protein that is similar to the protein in E. coli, then we will put a white box there on this line. Okay, and so the white spaces mean those are genes that are not present in these other organisms that are present in the mycoplasma, in the minimal cell. And we did this in 5 groups. So first we took the genes where we were really confident about knowing what the function, what the protein this gene made did. And you can see that this is almost fully filled in. And we did this at a very high stringency. If we loosened up the stringency of our comparison of proteins, it would all almost completely be filled. So for the genes, we know what they are doing. Almost all of these are present in all bacteria and even in you and me and plants and everything else. Next, we did it with genes, with proteins that we were pretty sure about we knew what they were doing, probables, or a little sure, putatives. And those, again, you see that for the most part, these are present in all of life. And even for organisms that we didn't know what the genes did, we might know that if they're generic, that the protein goes to the membrane, or it's a transporter, or it's a hydrolase for something. Those are generics, but there are also proteins that are unknown, where we just had no idea. And those are the ones we understand the least about. But still, what this says is that a large function of the cell is present, the minimal cell is present across all of life on Earth. Again, if we tightened up the stringency of this, if we loosened the stringency of our search, this would have a lot more color. Now, as I said, we were surprised that there were so many genes we didn't know what they did. And this is what we've been working on mostly for the last few years. Now, I would say that instead of 84, it's more like 78. I should have updated that for today. And I will point out that one reason there are so many genes of unknown function. So mycoplasmas, and we'll get to this, are evolving faster than other cells as part of their genome reduction. So the longer these lines are, this is a phylogenetic comparison, a dendrogram of all bacteria. And you see these lines are the largest, the longest for mycoplasmas. That means they are evolving faster than other bacteria. So a protein in a mycoplasma may have evolved so far from a comparable protein in another organism that they no longer look alike by simple blast searching. So that's an important point. Genes may perform the same function, but may no longer look similar. Okay, back up. I now want to talk about a paper that we published last year, 2 years ago, about, it's just amazing. We were stunned about, so this is Li Ji-sun, who's a geneticist, Kim Wise that I mentioned before, an MIT graduate student, James Pelletier, he's now a postdoc in Spain, and Elizabeth Strachowski, who's a physicist at the US National Institutes of Standards and Technology. So here you see 2 electron micrographs of first wild type JCBI SYN 1.0 and a cell that has the segment 6. It is reduced in one of the 8 segments. So segment 6 of the 8 is reduced. These 2 micrographs are at the same magnification. So what you see is these are 400 nanometer spheres, and these are all over the place in terms of sizes. This was just astonishing. Now, we decided we wanted to investigate this, and James and Elizabeth built these microfluidic devices where you have a channel that has about these dimensions, and through it flows growth, mycoplasma growth media with cells, a few cells in it. And every now and then, a few cells make their way into these tiny chambers you hear off to the side. And so you can see, the rate of flow. This is what the chambers look like. You get a few cells in, and then you can watch these over time, as you see in this micrograph, and you can see how the cells grow to fill the chamber. We have bright field and cytoplasm stained with M-cherry over about 12.5 hours. And here's what it looks like by video. Bright field, M-cherry. This is what we expected to see. It's exactly what we planned to see. I felt great about this. This was wonderful. Okay, and you can see that bright field, cytoplasm, DNA staining, and using a dye that stains the membrane. This is what we expected, again. Now, the next slide, I want you to prepare to be shocked. I'm going to show you a video of the minimal cell in one of these microfluidic chambers. This was one of the most astonishing moments in my scientific career. For those of you who are scientists or plan to be scientists, there are those days that you come into the lab and you see something that is so astonishingly different than the way you thought nature to be, that it just changes in an instant how you viewed biology. So here we go. Just look. One cell. What is this? It's a noodle. It's making these vesicles. What's going on? This is 13 hours of growth in a tiny little chamber. This is not how cells are supposed to divide. This is wrong. And we continue trying to figure this out. Again, bright field, DNA. So here you got one of these vesicles that is full of DNA. And sometimes we see vesicles with no DNA. And here's membrane stain. So we were trying to figure out what was going on here. And in our collection of cells that were built in the process of building the minimal cell, we had a cell that we realized looked normal in microscopy, called JCVI SYN3A. It has 19 additional non-essential genes in genome segment 6. So this includes a ribosomal RNA operon, the second ribosomal RNA operon, and then we saw 2 genes that we knew exactly what they did, 521 and 522. So 522, FTSZ is present in almost all life on Earth, a eukaryotic version of it or a prokaryotic version of it. And this is another CEP-F called a cell division protein. And we hypothesized that if we added these 2 genes back to SYN 3.0, that it would grow and function normally. We did that and it didn't. It still looked wrong. So then it took us 2.5 years and doing an elaborate set of hundreds of different mutant strains. We realized that we had to add 7 genes. If you added 6, it looked completely unusual, but if you added all 7, it would restore normal cellular function. And again, it's the 2 known cell division proteins and 5 genes that we don't know what they do. We're getting some ideas now, but still, we don't really know what they do. And so we realized that we may have accidentally recreated what cells looked like before modern cells evolved. So the original cells, and part of what made us feel this way, is we got a note from 2 paleobiologists from Poland, and they wrote my colleague Elizabeth Strzalski, and they said, congratulations on their article. We were surprised to find that minimal cell JCVI SYN 3.0, that's the one without these 7 genes that divide so strangely, gives a similar, if not identical, pattern of cell division that we see in fossilized bacteria that are 3.4 billion years old in rocks from South Africa. And this is what our cells look like and what their paper look like. And here's kind of what we think may be going on. So in all cells on Earth that we know about, you form cells get bigger, and then they form this cell division protein and lipid plane between the two daughter cells, and the cells divide. But our cells, they don't make this plane, which FTSZ is critical to that. So instead, we think our cells are dividing based on the physics of lipids, the physics of membranes, of bubbles. So here's a bubble, it gets bigger, and then you get two bubbles. And what we are now studying the process of cell division in these organisms to see if we can figure out what proteins are involved, what the physics of this, looking at membrane curvature, at surface to volume ratios. And this is something that I think you're going to be more stories about this over the next 2 or 3 years. From out of nowhere, Zan Luthi-Schulten, who is a computational biologist at the University of Illinois, wrote us saying that the minimal cell, and that was her graduate student standing behind her, the minimal cell is an ideal system on which to build computational models. They had been doing this with E. coli for years, but they loved the idea of something simple. Here's Zan again, and the postdoc who really did the key work that I'm going to show you about here. So Marion Brewer built a flux balance analysis model that took into account all of these reactions. In fact, it took into account 155 genes. And here we think of, for instance, the ribosome, which is 3 RNAs and about 80 proteins. So that just counts as one gene basically here. But 300 reactions, 300 metabolites. It is a complete network of the things we know about in the cell. And the reactions are largely supported by experiment. And in their model, the cell divides in 2 hours. And this is based on known reaction rates of the enzymes, the genes involved, the proteins involved in the flux balance model. And with this, you can design new experiments, and you can think about how to do things. So the computational models enable you to better design experiments. The model predicts something, we experimentally test it, and then the model goes back and you will try to do it better. So for instance, the model predicted that in order for the cell to divide in 2 hours, the cell had to have a transaldolase gene. Without a transaldolase gene, the cell would divide in 8 hours. But when we looked at the cell, there's nothing that looks like a gene that codes transaldolase. Similarly, the wild type cell has a pyruvate dehydrogenase. It's a 4-gene complex. We took away 2 of the 4 genes of pyruvate that makes the pyruvate dehydrogenase. And the model, the computational model, said the cell should grow 18% faster. Now, Andras Cook in my group put those 2 pyruvate dehydrogenase genes back into the cell, and he was very disappointed in the computational biologist because instead of growing 18% faster, it only grew 16% faster. That's a little joke. Andras also is looking at this transaldolase question. And so we did a gene complementation experiment. So this is the reaction that transaldolase does in the pentose phosphate pathway. We took a known transaldolase gene from Bacillus subtilis, and we installed it in the minimal cell in a landing pad that in the organism called Syn3B. And so we reasoned that if there's already a protein functioning as a transaldolase, TAL, adding a second transaldolase to the gene would make this quasi-essential transaldolase gene non-essential. And we installed the gene in the landing pad. We did transposon bombardment. And I should say that we have identified a gene that we think is the transaldolase. And now we're going to be testing that to see if this gene that looks nothing like a transaldolase does that reaction. And that would be really cool. So that would be taking a gene that we have no idea what it did, and now we're saying we know what it does. The minimal cell may also, again, recapitulate what primordial cells look like before the evolution of chromatin. So Ronald Rodriguez is a new postdoc in my group. Ben Gilbert is a graduate student in the computational group in Illinois. David Gonzalez is a University of California, San Diego proteomics person, and Remus Dame does chromosome conformational capture studies, 3C at Leiden University in the Netherlands. And so again, we just got lucky. By a genome minimization accident, it gave us a clue what cells may have looked like before the origin of chromatin. Chromatin being, so our cells, we have histones where our DNA, yours and mine, humans, is wrapped around these protein complexes called histones. Well, bacteria have a similar system. They have a protein called histone-like protein, and it's an essential protein involved in chromosome condensation. In E. coli, there are about 30,000 copies. In wild-type Mycoplasma mycoides, there are about 6,000 copies. Based on proteomics, we were stunned to see that the minimal cell only has 28 copies per cell. So this is what we think happened. The top is the list, shows a segment of the minimal cell genome, and you see here's HUP-A, okay? And here's the same gene in wild type. Now, GPSA, as evidenced by this triangle, shows that it is not essential. So we took this gene out of the wild type cell to produce the minimal cell, okay? And by doing that, what happens is by taking out GPSA 349, we lowered the expression of, so we have just this now, and it reduced the expression of HUP-A. We sent cells, we sent the minimal cell to a collaborator, Remus Dame in Denmark. And what he does is they do a reaction called chromosome conformation capture. And there you take cells and you use a protein to chemically cross-link DNA segments that are not immediate, like they are not contiguous on the strand, but rather you've got two pieces of DNA that loop together. And this is mediated by proteins. And so you then cross-link the DNA, fragment the DNA, and sequence it to see what pieces of DNA are next to each other. Now, if you do this with E. coli, what you see is that there are a lot of pieces of DNA, genes that are not exactly next to each other, but they are interacting with genes that in some cases are close, in some cases are farther away, as evidenced by how wide this line is. But in the minimal cell, it's just a straight line. We see that there are no long-range chromosomal interactions. So what we think has happened is with the absence of histone-like protein, there may not be any long-range chromosome interactions. And we imagine that this may be what cells looked like before they invented histone-like proteins. And it allowed for more control of gene expression and condensation of the protein. So what we're going to do is we're going to put gene 349 back in, and what we expect to see is much greater expression of the HUP-A protein, and then we'll do the chromosome conformation capture experiments again. Last week, 2 weeks ago, a paper came out in Nature about the evolution of a minimal cell. This is called adaptive laboratory evolution. So the one that came out in Nature was done by my group and these two guys at the University of Indiana, and this came out in Nature. And then in Press, there's a paper in iScience done by collaborators across the street at the JCVI, at the University of California at San Diego. And so the idea here is we're going to take the minimal cell. People were not even sure if it would evolve like a regular cell. We have taken away so many genes that it may not evolve the way mycoplasmas in general do. And I've already shown you that mycoplasmas are evolving faster than any cells on Earth, presumably because they've lost some of the genes that repair DNA damage. But for this experiment, the plan was, for adapted laboratory evolution, we're going to take a single colony, here's a fried egg colony, and we're going to take that and inoculate growth media in a small tube, and then we're going to split that big tube into 10 small tubes. And those tubes, we were going to grow them for 24 hours at 37 degrees, and then move a tenth of the media in those tubes into the same amount of media in a new set of tubes. And for the Indiana paper, they did this every day for 300 days. For the UCSD paper, they did it every day for 100 days. So with both systems, they also did wild type. All right, after 100 days or 300 days, what we did is we measured the growth rates for each of the 10 replicants of wild type and minimal cells. And we found the wild type division time went from 60 to about 50 minutes. The cells became more fit for growth in the growth medium. And think about, the cells had been originally discovered in a goat, and then they were not passaged a great deal after that, because people studying goat pathogenesis wanted the cells to be as similar as possible. So we find that if you grow them in growth media, with continuous culture, the cells, not unexpectedly, they have adapted not to live in a goat, but now to live in SP4 growth media. The minimal cell went from 120 to 100 minutes. Again, what that told us is it demonstrated in both studies that even after minimization, the cells evolved like a wild type organism. I wasn't surprised, but I was really pleased for this. Now, we also sequenced the genomes of each of the 10 replicates of minimal and wild type cells. And what we found is that there are a lot of genes there were a lot of places we had the same mutations in all or most of the 10 replicates in both wild type and minimal cells. So here you see, this is the UCSD study, and each of these columns represents one of the different isolates. But here you see so here we had I think it's 14 out of 15 had the same mutation was present, but in many cases, we got mutations all across the board. A lot of the mutations were subtle. They would be mutations in transposons. They would be mutations in the transcriptional regulatory sequences immediately upstream of genes. But this was an interesting story, and it again shows how the cells became more fit. Now, the Indiana and UCSD studies, as I said, they were a little different. In Indiana, they didn't shake their cells, and they put a pipette into the bottom of the tube that had incubated for 24 hours and sucked out the media from that. whereas in UCSD, they shook the cells and they just took an aliquot from the top. So what we found in Indiana when we did the sequencing as well as EM is that protein, cell division protein, if you remember from the cell size thing, the cell division protein was mutated both in wild type and minimal cell. And we put the same mutations back into the wild type and original minimal cells. And what that resulted in, the wild type cell diameter and cell size increased from 400 nanometers, to 800 nanometers, seen here, and the minimal cell decreased from 400 nanometers to 315 nanometers. This is something we weren't expecting, and I want to look into this, but it's an interesting story. Last little story before we wrap up. No, next to last. We want to build a living cell from non-living parts because it's never been done. The synthetic cell community would argue that we have made not a synthetic cell, but a semi-synthetic cell. And so what we want to do is build a cell from dead parts. So this is our plan. We're going to take Mycoplasma capricolum cells, the recipient cells in a transplantation reaction. We're going to grow a bunch of those. We're going to isolate the cells, disrupt the cells, and get the cytoplasm out. We're going to make a cell-free transcription translation system. We're going to put a plasmid into that encodes transporter proteins and a secretory system. Then we will mix this with lipids that are the same as a mycoplasma membrane, and spit them out as 500 nanometer vesicles. We will then put those in a tube with nutrients, and what we're going to hope is that these cells will now put proteins, transporter proteins, and a sex system in the membrane of these little cell-like vesicles. And what we hope is that will produce, we may have to do some funny things, but if we now add a genome to this by transplantation, by fusion, by whatever process, that will produce a living cell from non-living parts. And so we started this in 2018. And the first step was to make a cell-free membrane from the cytoplasm of mycoplasmas. Now, for 40 years, people have been doing this with E. coli and lots of other kinds of bacteria. You can buy these systems. They're great for making proteins in vitro. But we were completely unable, trying desperately hard with a mycoplasma to make a cell-free system. And we realized that mycoplasmas, for reasons that may be involved with their parasitic lifestyle, have an RNase on the surface of the cell. So we finally realized that you could take an E. coli commercial cell-free transcription translation system and put mycoplasma cells in it, and it would degrade the RNA in the E. coli ribosomes. And we were about to give up, but a Dutch graduate student had one more idea to try this spring, and he did something that they do in eukaryotic labs. He took the minimal cell, a lot of them, and put them in what's called a cell disruption chamber or a nitrogen bomb, where they put the cells under 3,000 pounds per inch of nitrogen pressure, and then they release the pressure to no pressure instantly, and the cells blow up. And from this, he was able to get a cell-free transcription translation, system. Back up. Transcription translation system. So now we're finally getting around to trying to build the cell that we want to do. Last thing I'm going to talk about. I'm sorry I've gone so long. The minimal cell as a possible anti-cancer therapeutic vector. And this is some work done first by my graduate student, David Brown, who was doing mammalian cell virology in our labs at the JCVI. And he was uneasy using his HeLa and other kinds of mammalian cells in the same incubators where I'm growing mycoplasmas. And I told him that I had been doing this for 20 years in my scientific career and never had a mycoplasma contamination, but David was nervous. And so he put the wild type cell in, and sure enough, it contaminated the cells. But when he put the minimal cell in, it just died. It couldn't live in the mammalian cell culture. And from this, we had some other ideas. We realized that it may be invisible. The minimal cell just might be invisible to the human immune system. So the US National Institutes of Health has a program where they are engineering bacteria, E. coli, Clostridium, Salmonella, other types, to express proteins that could kill cancer cells. And the cells are designed to seek out cancer cells when they are injected into patients or animals with cancer. Now, even if this works, the cells are alive and you can get other problems. The patients are now going to be infected. So we thought about how minimization might have altered the JCVI minimal cells' capacity to interact with the human immune system. Mycoplasmas are already really good at avoiding their host immune systems. They have no cell wall. They have low G plus C. Remember, bacterial unmethylated G plus C elicits a toll-like receptor response. That is one of the defense mechanisms our cells have. And they have minimal capsule. The minimal cell has already lost 80% of the presumably antigenic or potentially antigenic proteins on its surface as part of minimization, and it makes no capsule. So what we found is that if you mix the wild type cell with HeLa cells, you can see the wild type cell, if you stain them, here you got the light colored mycoplasmas on top of the HeLa cells. The arrows show this. But if you do the same thing with the minimal cell, you see it's dark. And so this said that our cells are not binding to the mammalian cells. And next, we mixed our cells with phagocytic cells, phagocytes, human phagocytes. And so if you do the wild type cell, what you see, it's hard to see because it's dark, but the wild type phagocytes are just gobbling up the bacteria like Ms. Pac-Man. But if you do the same thing with the minimal cell, it does nothing. The phagocytes don't even see them. So this gave us further evidence that the minimal cell is potentially just, we have eliminated its capacity to be detected by the immune system. This made us think, and again, this is shown here in studies that you see the wild type cell is detected by the phagocytes, and the here's controls that are not phagocytizing, and the minimal cell is hardly seen at all. And this is about the same also for polystyrene vesicles of the same size as the minimal cell. So this got us to thinking. The mycoplasmas are gobbled up. The minimal cell isn't. What could we do with this? So imagine we take the minimal cell and we engineer it to express a protein on the cell's surface that could kill a cancer cell, something like TNF-alpha related apoptosis inducing ligand, or TRAIL. And this is used in CAR T therapy for cancer therapy. And we have that on the surface. And then we'll also engineer a single chain antibody that would bind to, say, colon cancer. All right, so now imagine that we've got a patient who has, sadly, colon cancer. I had colon cancer 2 years ago, so it's near to my heart. And we're going to inject those cells into the patient, and hopefully, we're going to do it in rats and animals first. Hopefully, the cancer goes away. And as I said, we're getting ready to send these cells to be tested at the University of North Carolina. But this could be so much better than some of the current approaches for cancer therapy. We're going to try it. This could be fantastic. with that, I'm going to wrap up and say that, the minimal cell, this is the most malleable organism on Earth. I can manipulate this organism in ways you cannot do with yeast or mammalians or coli or anything else. We have a defined growth media that's reported in the literature. We know the functions of most of the proteins, a larger fraction than any other cell on Earth. The cell is easily visualized in electron microscopy. We have a huge community of researchers working on this, and we've eliminated its pathogenicity. So what we want is a tool to investigate the first principles of biology. Our vision is we'll have a cell where we know what every gene does, every atom does, and we're developing computational algorithms that will let us build organisms to devote maximal cells energy to their purpose, and eventually redesign all kinds of cells to solve human problems. I will close in saying the mycoplasma community, and I know there are a dozen labs, I think, in Japan that are part of this. We have had the last 2 years in September virtual workshops where different students and postdocs and faculty from different groups give brief presentations to people all over the world. They've been wonderful. And the next one will be this year in September. We'll probably do it over 3 several-hour sessions. And write me, and I will send out notices to lots of people about this. And of course, it takes a huge number of people to do the work that you've seen from a lot of different places. I should have highlighted that the discovery that the minimal cell doesn't affect mammalian cells. So I was also a part of a study done by Itaro Yamagahara from Osaka Women's and Children's Hospital that gave us a lot of ideas, and it's a great collaboration. So with that, I'm sorry I went so long, but if you've got any questions, I would love to try to answer them.
Yes, thank you very much, John. So then, we are very happy to see that you are talking very, very actively. So then, the people say that something like that to me, but you don't look the patient. You look very healthy.
I am healthy.
Yeah. Okay. We are very happy. Yeah. Really. And also that you show the really, the nice, the results. So then I'm also really, excited. So now it's time for the discussion from the floor. Hi, Hidemin, go ahead.
Can you hear me, Dr. John Glass?
Hi, John. Can you hear him?
This microphone.
Yes, I can. Very well.
Thank you. Thank you for your great talk. It was very easy to understand for me, a non-native English speaker. My question is relating to the method for constructing minimal bacteria. I'm curious about if there any possibility to occurring recombination between Capricolum original gene and newly introduced synthesized gene before the recipient cell division?
Well, we were worried about that. And first of all, we knew that Mycoplasma capricolum and Mycoplasma mycoides, neither of them perform homologous recombination. And when we sequenced the genomes of the transplants, and compared it with the genomes of the Mycoplasma capricolum, we found no evidence of any recombination. Of course, we couldn't, if you got like 5 or 10 bases that recombined, how would you know? But there's no reason to think that. And remember, we are not taking genomes. Well, yes, I think I'll stop at that.
Thank you.
Yes.
More questions? Yes, please go ahead. And if you have the, additional more questions, so you come to the microphone and in line, Hana-chan, maybe that you come to be ready.
Thank you for your great lecture. So I'd like to ask a question about the function of the JCBI-C3 genome. So if the cells of multicellular organisms expression the function of the JCBI3 genome, is it possible to make single cells live in a single cells, live like single cells?
Well, so these cells are single cells. They are not multicellular. The cells will clump together, but it is not like it's not like a Volvox or some of the small, very small eukaryotic organisms that are multicellular. These are single cell organisms. They stick together. Lots of bacteria do that. A colony is not one organism. It is millions of organisms that are just in one place, and they may have some community organization, because clearly the mycoplasma colonies, they don't just form a pile of cells. They have a specific size and shape that is not just a pile. They look like a fried egg. Does that address your question?
Oh, okay. All right. Thank you very much.
Yes, thank you.
Yes, Hana-chan, please go ahead.
Hi, thank you for great talk, Professor John Glass. I'm Hana. I'm Hana from Makoto's laboratory. And before I moved to...
I am a big fan, Hannah, of your paper.
Thanks very much to introduce our paper in your great lecture. I'm so happy. And I have a question about transplantation of mycoplasma mycoides to capriculum. Transplantation is a most important key to make synthetic minimal cell. So, but in my understanding, it's still difficult in other bacterium, even mycoplasma to mycoplasma, if the phylogenetic tree is far. So it's this kind of transplantation is a special special for mycoplasma mycoides to capriculum, or is there a clue, key to understand it makes possible to in other bacterium in minimal cell? Can we identify what is a key to transplantation by using minimal cell?
That is a very, very good question. And it is something that we have been worrying about since we first developed the first transplantation reaction. As Hannah said, we have not been able to transplant anything but mycoplasmas in a small, closely related group. We have, and no other organisms outside of this set of organisms that are very closely related. The efficiency of transplantation drops off as the mycoplasmas that are closely related but get, so we looked at, I think, 6 different species that are to different degrees related. And the farther the two species are apart phylogenetically, the less efficient the transplantation. So just this year, I got a grant from the United States National Science Foundation, where we finally have money that we are going to try to learn how to transplant other kinds of bacteria. So to transplant E. coli genomes, or Haemophilus genomes, or Bacillus, or Synechococcus. Now, the lessons from mycoplasma tell us that we probably couldn't transplant a gram-positive genome into a gram-negative recipient cell. So what we think we are going to have to do is transplant not from one species to a different species, but from one species to the same species. So imagine a world where I can, and I think this is not too far away, where I could design a Bacillus subtilis genome, and I could send my design to a company like Twist Genomics, or Telesis Bio, or IDT. I'm sure they're probably Japanese companies that synthesize DNA. And a few days later, you get a yeast cell back that has your synthetic genome in it. And then you could transplant, but I would take a Bacillus genome and transplant that is not the same as wild type, but radically different. And I would transplant that into Bacillus, so that I would make a new strain of Bacillus just based on my design, as opposed to having to make hundreds of mutations, perhaps. So the way we're going about this, we have an idea about how transplantation works, but we're not really sure exactly how it works. We know what's necessary, but I'm hoping that a year from now, we will be able to tell the world how to do this. We're going to get there. I cannot believe that I am either so smart or so lucky that I picked the only organism on Earth that I would be able to transplant. I just can't be that good. Our team, I think we're great, but no one is that good out of all the bacteria on Earth that we got the one that will work. So I think we are going to solve this. Great question, Hannah.
Thank you very much.
Other questions? Yes, please go ahead.
Thank you for the interesting talk. I'm Shuji, a faculty member of Osaka Metropolitan University. My question regarding minimal cells. I'm not familiar with the cells, so I'd like to know the present situation of minimal cells. So at first, I assumed that it's quite difficult to define the essential cells, essential genes, because it depends on the situation.
Yes.
But it seems you could solve the problems, and then since generated Syn 3.0 cells. In this case, is it possible to use the JCBI Syn 3.0 or list of you generated minimal cells to produce any kind of chemicals, or we still need to design cells from the beginning based on the situation to apply for the minimal cells to produce any kind of chemicals?
So, Shige-san, that is, no one's asked that before, ever. And I think it's a really important point. So I think that our minimal cell, you could certainly build new pathways in it. For instance, Tobias Erb at the Max Planck Institute in Marburg, Germany, built a novel carbon fixation pathway that involves 17 different genes from 9 different organisms. And we are building that into the minimal cell to see if we can have the minimal cell perform this complicated chemical reaction. There's no reason to believe that it won't. Now, granted, I will have a cell that can fix carbon, but the media for the minimal cell, so the way we make it at commercial prices for the ingredients, it is $286 a liter. I'm sure Makoto-san knows well the cost of his media. Although we have developed a completely synthetic media, I haven't priced it out yet, but I'm sure it is hard to make, but I think it will be much cheaper. But our cell grows slowly, and so that would make it not ideal for product production. But there are industrial groups that have taken our minimal cell and put into it pathways for the production of some drugs. And what they wanted to use is to understand how these new enzymatic reactions that were added to a cell that performs sort of the basic functional requirements of life, do any of these newly added enzymatic reactions interfere with basic cell biology? And so for that, I think the minimal cell might be really good.
Thank you.
Yes, please go ahead. Kanamori-kun.
So can you hear me? I'm a newcomer of Professor Miyata's laboratory. So you indicated that the SYN3 can be anti-cancer therapy vectors. Regarding this, so I wonder that can SYN3 survive in the blood? Can SYN3 survive long time or so replicated normally?
I have not done the experiment, but based on its inability to bind to any of more than a dozen different kind of mammalian cells that we have tested, I don't think that it would be able to survive more than a few hours. But we know the genes that we could add back to it that would let it last longer if we wanted that. We also know that as part of the genome minimization, we have made the cells much more sensitive to antibiotics. We have gotten rid of transporters that would export antibiotics. But again, we know how to make the cell able to survive, at least in mammalian cell culture. Whether it would survive in an animal, I don't know, but we will be doing those experiments in mice later this year. Thank you.
Thank you.
And
Thank you so much
on joining Makoto-san's lab.
I was so glad to join Professor Miyata's laboratory. Thank you.
Thank you, my honor. And other questions? Yes, go ahead.
So thank you for great talk. I'm Daiichi Takahashi, a postdoctoral researcher in Makoto's lab. So my question is, minimum cells are cultivable both in liquid media and also form solid media, so then it can form colony. But when we see kind of the bacterial kingdom out further, the prokaryotic domain, there are a bunch of species that cannot be formed colonies, but they're still alive. So then my question is there is it possible to further remove genes only related for colony formation and create minimum cells, which is only available in liquid media?
Possibly, and we might, and the way we did our experiment, if those were, remember, the first thing we did is we required that the cells be able to make colonies. And so if we had done our original experiment a little differently, we might have knocked out genes that were required to make a colony. But because I don't know the genes that allow the cell to make colonies, we may figure it out accidentally at some point. Or if you guys have a hypothesis about this, it's something that we could try to do with you or teach you how to do on your own. But I think it is entirely likely that we could either make a cell that couldn't make a colony or couldn't make a fried egg colony.
Thank you so much.
Thank you very much.
Other questions? Yes, Asan, please go ahead.
Dr. John, thank you very much for the great talk. I am Ari Essen, and I'm a first grade PhD student with Dr. Makoto Miyata. And I have a weird question, maybe it would be sounding odd, but since we are transforming prokaryote genes, for example, our gene of interest, and we are transforming in the Syn3B cell, somehow have you ever thought, or maybe it is my weird question, that we can use Syn3B cell for transformation into the eukaryotes? For example, we want some of our gene of interest, or we want some our traits of interest, and we transform using this cell, and we can get that trait. Do you think so? Is it possible?
Well, keep in mind that, first of all, the mycoplasmas use a different genetic code. That would be problematic if we were to put the minimal cell genome into a eukaryotic nucleus, and it would be a different genetic code, and then so eukaryotic transcription and translation signals are different than prokaryotic transcription and translation signals. So I have a project here where we are building humans, we design and build completely synthetic human artificial chromosomes. And my collaborators have designed fully synthetic centromeres. And we put a variety of eukaryotic genes that come to about 100,000 base pairs. And if you make a human artificial chromosome, that is only 100,000 base pairs, it doesn't work. But we took their synthetic 100,000 base pairs and put it onto a minimal cell, we put it onto a JCVI Syn 1.0 genome. That's about 1 million base pairs. And now it functions and divides like a mammalian chromosome. It doesn't, to our knowledge, produce any of the bacterial genes, but it does function as a chromosome. I have another collaborator who is trying to take the JCVI minimal cell and have it become an organelle in yeast cells, where it provides something that the yeast needs in order to live. But again, that's putting the whole bacterium into the yeast, where it would just live there, and it might produce some of the bacterial proteins in the yeast. And so these are things that might possibly be done.
Yes, that's pretty right. Maybe in this way that you have told that you have the collaboration and that is working on, maybe we can use this kind of cells to in order to overcome the problem, for example, the rare blood groups or that kind of things, we may use that as well.
I don't quite understand that, but can you explain a little more?
Yeah, I mean to say, as I have said, that we can use them for transformation for eukaryotic cells, or maybe in that way we can use these kind of cells to make some synthetic blood products, or in that way.
Possibly, in the sense that you might be able to get bacteria, the minimal cell, for instance, that is a little smaller than blood, but if the immune system didn't care about it, you could put these in to be, human living in humans, doing all kinds of beneficial things that other kinds of microbiome therapies are not able to do. In theory, you could make the minimal cell, or the minimal cell, like you say, function as a human red blood cell, perhaps.
Yes, I got it. Thank you very much for the $1 million answer. Thank you.
Now, don't ask me how to do that yet, but why don't you do that? The Miyata lab, I think that would be great.
Yes, other questions? I have a question. So then, how often do you have discussion with Dr. Craig Venter?
About once a month.
Oh, really?
Mhm.
And you discussed also the details of the data.
Sometimes, probably that's a little less often.
Yes.
I have been given new administrative responsibilities at the institute.
Yeah.
So.
And also, I'm a little bit curious about the way to determine the destination. For example, so in our usual lab, so then usual, what is usual anyway? In our labs, so then sometimes the young people that decide and search the small possibilities by just their ideas. So then, how about the big project? So they always they need to have for example, the contract or something. So then start the project. How does we can't imagine it. So then how does it look like?
So with the original production of the synthetic cell in 2010 and then the minimal cell in 2016, we had large grants, first from the US Department of Energy of about $10 million, and then another $30 million from a company that Craig founded. And that got us to the point that we first built the cell. And there we had about 25 people working on this, trying everything we could imagine to build these organisms. Since then, I am getting exclusively funding, mostly now from the US National Science Foundation. And these are vastly smaller grants. So to build the original minimal cell, it was about 250 human years of work and $40 million. And I can't imagine getting that kind of funding for this project now. Whereas now I have one person who is and part of a technician who are dedicated full-time to the minimal cell, and I get $1 million grants for 3 years from the US National Science Foundation or from other government organizations. And I have 7 different kinds of projects going on in my lab, some involving the minimal cell, some things completely different. And always the student, so I have, JCD doesn't have students, but I have some UCSD students who work with us. But I have postdocs who do the main thing that we promised the granting agencies to do for the work. But then we can also do other things that we think might be interesting or worthwhile at the same time.
Yes, thank you very much. It's really, important and interesting information for us. Yes.
Yes.
So then, yes, thank you very much. So then now that if we don't have that, you may have more, but so then if you especially the younger people, if you have any questions that you give me and I made, forward to the John anyway. So then now we are closing our, really special seminar or the lecture. So then we again, we present great thanks to the Professor John Glass. Please go ahead. Yeah, thank you very much, John. So then now the 8 o'clock or 9 o'clock in Pacific time.
Yes, it's just 9:01.
Yeah.
Makoto,
So then,
I would very much like if perhaps we can arrange that our labs have a joint meeting every 2 or 3 months.
yes, great. Yes, so because we are, our major, the subject is sensory. Yes.
But you do spiroplasma and other mycoplasma work, and you guys do, and I think this would be fun.
Yes, I agree with you.
As long as I don't have to learn Japanese.
Yes. Yes, it's the very for us that that we keep we need to keep the English conversation skill for all members, so that it will be the really great help. Yes, thank you very much. Thank you. Yeah.
Thank you.
So then,
Domo arigato.
Yeah. ありがとうございます。Yes, see you.
Bye now.
Bye. Yes, this video would be the treasure of us. じゃあ皆さん来ていただいてどうもありがとうございましたんで、うちの研究室の人たちはちょっとお片付けしてくださいね。まあ、そんなたくさん人数いらないけどね。で、授業の人たちはちゃんと感想を書いてね。はい。それで、授業で関係なく来てくれた人たちも本当にありがとうございます。以上です。